Oh boy.
Hey, hello. Good afternoon, everybody. Today is my pleasure to introduce Professor Checo Colón-Gaud. Checo is professor in the Department of Biology at Georgia Southern University. He has a PhD in zoology from Southern Illinois University. He worked with Matt Wiles. And Checo does really cool work on aquatic food web structure and the role that consumers play in processing organic matter, and in turn, how this processing influences, ecosystem functioning. He has studied a range of aquatic ecosystems, coastal plain streams and wetlands, and tropical headwater streams. Additionally, on top of his science, Checo has a really impressive track record of advancing diversity, equity, and inclusion in the freshwater sciences. He has led the INSTARS program at the Society for Freshwater Science. He has coordinated a number of programs that provide professional development and networking opportunities for underrepresented minorities. And the last development on that is that he just got this large NSF grant with partners at Berkeley to advance this mission. Particularly, they will train students to make sure that research opportunities turn into jobs for them. So we are really excited, the freshwater folks and the broader department, to have you here. So thank you and welcome.
Thank you. I'm excited to be here as well. Do you want me to start sharing? And what I'll do is, once I start sharing screens, is I won't be able to see as much. You just got to let me know that, let's see, hold on one sec, that I'm in the right screen. Let's see. All right, you could all see my screen. Give me a thumbs up.
Yes.
Yeah. All right. Well, I'm super excited to be here, and not just because of the partnerships that we've started that include folks out in Berkeley, but also because this is a great opportunity to see colleagues and see students that I've interacted with in your programs and get to find out a little bit more about their progress and their work. But also because this is kind of like the nice thing about being in this type of position, that you get to talk and tell stories about the type of work that you do. And I want to emphasize particularly that, telling stories, because I've structured my seminar maybe a little bit different. The folks that know me out there know that I'm a bit of a storyteller, and being that it's past 6:30 here in Southeast Georgia where I'm at, I'm mainly going to be telling stories, and hopefully they'll land at what my philosophy is for work and for research and mentoring, and hopefully expand on the opportunities that we have available. I also added here my contact information because I tend to be long-winded, so I just want to make sure, just in case folks are starting to trickle or want to ask me about things later. My email address is always good, and then I've added my social media. I've been learning to interact a little better with social media as we get into some of these projects. But to be able to tell my story in terms of how I kind of landed at this track as a freshwater scientist and the type of work that I do today and the type of work that inspires me, I got to kind of start a little bit at the beginning. If you haven't noticed from my name or from my accent, I'm Hispanic. I grew up in the island of Puerto Rico, a tiny tropical island in the Caribbean. And that just kind of got me excited about water because I was surrounded by water all the time. I don't know if you can see my cursor, but I'm out there in the city of Mayagüez, which is in the west coast of the island. And I grew up most of my life around like rivers and around the beach and the ocean. And that really kind of inspired me, maybe in a way that I didn't think about academically, about the type of work or the type of scientist that I wanted to be in the outdoors. However, I went to school at the University of Texas at El Paso as an undergraduate. I left my tropical island and moved out to the Southwest. And I really got exposed to very different environments. And I do want to mention my undergraduate movement primarily because I went to a minority serving institution. As somebody whose English is a second language, I think it was important for me kind of getting exposure to go to an institution where I was really surrounded by a lot of Hispanics, a lot of Mexican Americans. I think at the time I was there, the population of students at UT El Paso was about 87% Hispanic. So it really gave me a nice transition into the culture of being a college student and getting exposed into work. But also leaving a tropical island in my tropical humid environment, I was really exposed to a very different ecosystem in the southwest of Texas. And that was exciting too, because it really got me to think about just different types of diversity, different types of environments. And it started to get me hooked in environmental science. I got some of my first chops out there in terms of doing research, as you see pictures of me out there in the desert and pictures presenting as an undergraduate. But I was very fortunate that I had a good mentoring team out there, or a group of professors that encouraged me to pursue research. And some of those first opportunities that I want to touch on as well were as a NSF REU fellow, or research experience for undergraduates fellow. So towards the end of my undergraduate degree, I applied for the summer internships. I know many of us are familiar with obviously the NSF and the REU program. There are many REU programs, and I know many of you graduate students might have participated in some. But for me, this was my first exposure to something like the National Science Foundation. And I don't take it for granted now, because as I talk to students and I tell them about the NSF and I tell them about the type of work, particularly like the work that is done at the long term in this research sites, that is really important to the pathway that I took as a scientist. So as an REU, I was able to spend time in Kansas at the Kansas Prairie Biological Station, which is an LTER site. And now I had a chance to work in aquatic systems again, but in these prairie streams that were beautiful and obviously very different, not just than the sites that I was used to in Puerto Rico, but also the sites that I was exposed to as an undergraduate in Texas. And what you see me out here is I'm playing in some of these prairie streams. I had the opportunity to work with a great group of mentors. At the time I was there, I worked with these two gentlemen. The gentleman on the top left is Dr. Matt Wiles, who is now at the University of Florida. And on the top right is Dr. Walter Dodds, who is at Kansas State. And they were a great group of mentors for me as an undergraduate, coming from a minority institution with very little exposure to field sites and to sites that were part of this large network of ecological research sites. And I got to mention that mentors do matter, because even today, both Matt and Walter are important mentors for me, even as a professional and as an academic. So I want to touch on that very early in my career, I had exposure to these mentors that still play a role in the type of science that I do today, but clearly in the type of philosophy that I have for mentoring. Now, while I was out there in Kansas, I got to work with chironomid midges, with true flies, larvae of this great organisms, and got a little bit of exposure to their life history. So what I'm doing there, as I'm kneel down in the stream, you see that I'm playing with these plastic boxes. So those were practically midge hotels, if you could think about them that way. So they're growth chambers where I was measuring these chironomids, and then I was placing them in this environmental, these chambers in the stream, and allow them to grow. And over time, I'll come back and I'll try to pull them out again and measure how much they have grown, as a way to quantify their biomass, but also their growth of mass over time, or their secondary production. So I got hooked on not just aquatic insects, but really trying to understand the role that they play in the system and an ecosystem function. So I was trying to understand how the growth of mass really became critical as either a food source for other consumers, or in terms of the roles that they play in the environment, particularly organisms like dipterans, or the true flies, that if they do grow enough to become aerial adults, they're transferring a good amount of energy to the terrestrial system. So I got this time to spend in Kansas, and not only did I get exposure just to general ecology and things like I mentioned now, like the life history and secondary production of consumers, but I really tried to started to kind of get my interest in food webs and the role of disturbance. This is a picture of one of the prairie streams out in Kansas Prairie, where during the summer I was there, you would get this very quick flash floods that would very quickly turn your chironomid chambers or your projects into zero, and you had to start over if things got washed out. So I got an appreciation for the role of disturbance in field work and field science. And then in terms of my already interest in water, I started to get some of the first exposure to theoretical frameworks in stream ecology, like in particular, the river continuum concept, or the RCC. For those of you that might be aquatic students or aquatic faculty, you know that this is an important concept, an important piece of work in our work in river ecology. But more importantly, if you think about the RCC and the systems that I was working on in prairie streams in Kansas, they don't necessarily like fit the picture that you have in your mind for rivers along a continuum where the headwaters are very forested and you have these trees that contribute not only a lot of leaf and organic matter, but also serve as some sort of shade or block to limit light penetration and primary production. These prairie streams, the headwaters are really surrounded by grasses, so they're almost flipped. They're almost behaving more like middle reaches. Now, still fitting the framework of the physical driving the biological in river ecosystems, but not necessarily the poster child or the picture that we imagine for the river continuum. So very excited from completing my work as an undergraduate, which actually turned into publications, not just for my mentor, but also for the graduate student that I was working with. And I mentioned that as being important because in these RAU programs, that peer mentor system and the role of graduate students as really near peers to undergraduates is very, very important. So I'll plug that for any graduate students that are in the audience and have the opportunity to work with undergraduates. So I took my experience and you know, I was packed with excitement. And I went to pursue a master's in fisheries at Louisiana State University. And I'll mention this because my experience here is in a little bit more of an applied system. My master's degree is in a college of agriculture. We're really looking at problems or questions that were somewhat more applied. But more importantly, it exposed me to very different ecosystems again. I'm no longer out in the Southwest desert. I'm no longer in the small prairie streams of Kansas. Now I'm working with very large rivers in the south, in these very large floodplains, and partly in the coastal plain, but also really exposing me to more theoretical frameworks in river ecology, like the flood pulse concept, and the importance of these floodplains in not just providing habitat for spawning and for foraging of many consumers, but really places where a lot of activity happens as organisms may move into these pit stops before they exchange energy with the main channel or the corridor. But it also kept my interest in food webs, maybe at a much larger scale, although I'm trying to like paint quite a reach here if you think about my aquatic invertebrates to perhaps like contributing to the energy or the food or the transfer of energy for things like alligators. But that was the mentality, really thinking about how the small organisms play a role in transferring biomass to much larger consumers or to larger trophic compartments. Now, while I was out there, and you know, you don't have to adjust your screen. This is a very old picture, probably taken with a disposable camera. I had a chance to work on an exotic macrophyte and the invertebrates that were colonizing beds of hydrilla, which is an exotic that has gone widespread throughout the South and through most of Texas and even across all the way to Florida. But I had the opportunity to really get back into a different type of disturbance and really think about how these exotics might be removing native macrophytes and changing the habitat or changing the dynamic of those ecosystems as they clog some of that material. And that's me just standing in a bed of hydrilla that I could stand out there and not even necessarily get out submerged towards the swamp or the water. So while I was out there, I had a chance to develop a sampler. That's the picture that you see in the right, which resulted in some of my first scholarly work, which was exciting too as a master's student to really contribute to the literature. And I try to plug this one too, because the one on the right was my first publication as a graduate student, and not necessarily just for the publication, but I like to say that my first paper was published in the Journal of the Kansas Entomological Society with a great acronym of JOKES. So I was a heck of a comedian at the time. But it really exposed me to differences in terms of thinking of our work in a more applied setting. I knew now that the work that I was doing had the opportunity to contribute not just to, you know, the natural ecology or the ecology of the system, but really to contribute to what we know for commercial and even recreational fisheries. And some of the work that I was doing already was kind of getting me towards the way of trying to understand how energy was transferred from small consumers like invertebrates all the way to larger consumers like fish and amphibians and so on. So I took my experience there to my PhD at Southern Illinois University, where I worked with a great group of scientists, and I wanted to plug this as well, because at SIU, as I was working in this tropical amphibian declines in streams project, I got a chance to, there's me right here in the back, putting bunny ears on my advisor, but I got to reconnect there again with Matt Wiles, who was my undergraduate advisor as an REU. So if I try to think about the impact that mentors make in your career, as an undergraduate, I was not just inspired by the work that Matt was doing, but was able to reconnect with him and learn from him again as I was trying to develop into my own scientist into a PhD. But the other thing that I want to mention out here is that in this project, the opportunity that I had was also to work with a very diverse group of scientists. In this group, which actually, other than Matt Wiles here and Karen Lips, who were the PIs of the project, Matt is now University of Florida, Karen Lips is the University of Maryland. All these students around me are PhD students that are all now faculty at different institutions. Dr. Roberto Brenes is at Carroll University, Dr. Mason Ryan at the University of New Mexico, Dr. Misha Hunt at Drexel, and Dr. Scott Connolly at UGA. So I was really kind of expanding on this network of people that were not just like-minded and excited about working in streams, but people that are now colleagues and mentors of their own. Now, this project was exciting too. We had a chance to work in Central America as this wave of amphibian declines was just running through that entire part of the land. And this is probably something that you've seen on the news or heard about, but this disease-driven declines due to chytrid fungus were really wiping populations of stream-dwelling amphibians, not just throughout like North Central America, but have moved almost in the same predictable fashion. And also another wave that has moved from South America almost to meet some of these areas between Central and South America. So while we were out there, we had a chance to study sites that have already experienced amphibian declines versus sites that at the time that we got there still had healthy amphibian populations, but over time we knew that predictably those were going to be in demise. Now that was exciting as not just as a graduate student. I mean, I'm going back to the tropics, I'm going back to what I was used to, but also got a chance to really experience some incredible diversity in now a completely different group of consumers. And amphibians, a lot of this rainforest and tropical frogs that not only had beautiful colors, but also beautiful sounds, and it was a sight to see. But knowing that many of these, particularly the ones that were associated with water, associated with streams, were in a lot of trouble. So to think a little bit about the work that we were doing out there, this is one of our streams in central Panama. We want to understand what the role of amphibians may have been in these systems. So when they're in the riparian zone or in terrestrial areas, not only do they look incredibly adorable, but they're also playing an important role as they feed on a lot of emerging insects, a lot of small consumers that might be now interacting or exchanging energy from aquatic habitats to terrestrial habitats. But they also become food themselves. They become the prey for many snakes that target that target frogs around streams, but also birds and mammals. And again, I mean, they were now a food source too for some of the consumers that you expect to see along this riparian areas. Those of them that didn't necessarily get eaten, many return back to the stream and deposit energy-rich eggs and energy-rich immatures back into the aquatic system, now exchanging energy from the terrestrial into the aquatic. And in the aquatic system, they played important roles as grazers, was the scrapers or primary consumers of a lot of algae and a lot of like that production or primary production that you expect to see in the rocks and a lot of bioturbation in terms of sediments that accumulate in this system. So we were excited to think about the possibility that the major consumer that is keeping some sort of top-down control on primary production was likely going to be removed on a nature playing an experiment or manipulation in the systems for us. Now, if I take you a little bit down or underwater, you could see that these tadpoles were not only super abundant. I mean, they had high densities. I'll help you out if you can't see them. There's a bunch of tadpoles out there in yellow, and I'm sure I missed some and some fish as well. But what I really want you to take from this picture is how these rocks, you have those patches of very green material, like that very green algae and primary production and moss might be growing in those rocks, that when you remove the tadpoles from these systems, visually you can already tell that the system had changed quite a bit. You have these very green patches and all your tadpoles stuck to those rocks, but then as you remove those tadpoles, and this picture happens to be from the same site a year post decline, you see that there's a lot more sediment accrual. The system itself, or like that resource base, has changed. And that's what was, again, a great opportunity for us. Obviously, quite a shocking opportunity to see your ecosystem change, but an opportunity to really understand the ecosystem level changes and responses, not just in things like the resource base, your algae, your primary producers, but in my sense, what I was working in this project was the macroinvertebrate community and some of those secondary producers and how they might respond. So to show you a little bit of data or a little bit of the work that we did there, this is just a very simplistic flow web or very simplistic food web. And what I'm trying to show here, and I'll have 2 slides of it, is what the system might look like in terms of other grazers or other primary consumers in the system in the presence and absence of tadpoles as the bulldozers or as the grazers and scrapers in the system as well. So what you see on the top are aquatic insects, and again, and what I have is 4 groups, 2 genera and 2 families of mayflies. Here we have algae at the very bottom, and now all those estimates that I'm showing, either the arrows or the numbers beneath the names of the invertebrates or even the algae, that I'm quantifying that as a flux or as a production or a movement of material or movement of biomass. So for example, at the box at the very top in blue, the numbers underneath those invertebrates, these are water penny beetles. This is a Lepidoptera and Petrophila, so moth larvae or moth nymph, and then 2 families of mayflies, leptophlebioids and batids. This numbers out here is the production in grams of ash-free dry mass per square meter per year. So a dynamic measure, a measure of how much mass those organisms are generating or turning over time per unit area. So the amount of biomass, heterotrophic biomass or consumer biomass that is being generated. In the box of algae, that's the amount of primary production that is estimated that those tadpoles will be, again, scraping or keeping top-down control on. And now the boxes and arrows really indicate the amount of consumption that you expect will be necessary for those invertebrates to reach that level of production. And what you see out here is, again, you know, that there's variability in the amount of consumption that will be needed or estimated based on ecological efficiencies for those different groups, with the larger or thicker arrows indicating a larger amount of exchange or flux of that material. So that's what we had when tadpoles were present, and you could assume that they're in some way interacting, either competing or facilitating that resource to other primary consumers. Now, if we look at the sites once tadpoles were removed or tadpoles were extirpated, the first thing that we notice is that it was an approximate 5-time increase in the amount of algae. Now you don't have tadpoles out there scraping and now keeping some top-down control in the resource, but now you have this increase in algae that you assume results in differences in the way that they exchange or are able to consume the leftover material. So what we found was that some of these groups, particularly of the major insect grazers, the water penny beetles, Sphetus and the Lepidopteran, they actually decreased, not just in their production, but also in the amount of consumption of the material. The water penny decreased almost, you know, to 50% less than what it was producing and consuming before, and Petrophila even more. However, the mayfly groups, they actually increase in their consumption of the resource. So now you have an exchange in terms of like organisms that might have been benefiting or in a sense like being facilitated by the way that tadpoles kept top-down control on that resource and provided perhaps a different resource that could be more feasible, more palatable for some consumers, whereas some other consumers might be able to now benefit from the change in the resource, which included perhaps some large filamentous algae and some other types of primary productivity. So that was exciting for us because we could tell that in these systems, as you're removing a major consumer, the system is not collapsing, but it's obviously changing, and it's changing in the way that energy was being transferred. And obviously it was exciting for us. This work resulted in, still results in a lot of scholarly activity and a lot of baseline data for work that continues now to try to understand how the systems are responding now, more than a decade plus of experiencing this amphibian declines. So this was great, and this was exciting work, and it provided new opportunities. As I finished my doctoral degree, I had the opportunity to participate in an NSF-funded postdoctoral opportunity in Puerto Rico. So now I had the opportunity to interact with another mentor, Dr. Alonso Ramirez, who's a collaborator as well. And this is us here in one of our Puerto Rican rivers. But if you think about it, I mean, this opportunity now brings me back home. And when I left the island as an undergraduate, I never imagined that the type of work that I was doing, that I wanted to do, was going to give me the opportunity to work back in systems that I was familiar with growing up. So I returned to Puerto Rico, and I had the chance to work at the Luquillo Experimental Forest or the El Yunque National Forest. If you had a chance to travel or visit Puerto Rico, this is in the northeast side of the island. As you could think about this as the tropical rainforest site for the NSF Long-Term Ecological Research Network, or the Luquillo LTER. So again, a tropical rainforest that you can imagine now has all these characteristics that we like to study to try to understand how these systems might be changing. And I want to highlight, if you look at, again, where it's located in the island and what the site looks like, here's a satellite image that I want to kind of touch base on, because this LTER site has really become an island inside the island. And when I say that, is because you can see the level of development and the level of just the urban sprawl from San Juan, the metro part of the island. Here's the water, but you can think about this being an isolated patch now of protected land. Now, the neat thing about this is that while we were in Puerto Rico trying to really generate excitement about understanding their food webs, these systems are actually dominated by crustaceans as their main macro consumers, and not just in a diversity of shrimps, but also some crabs. And they're not only taxonomically diverse and exciting, but they're also very functionally diverse. They do many different things. And the work that I was trying to develop in the island was very similar to what we were doing in Panama for the Amphibian Declines Project, but really to try to understand their food webs and try to come to generalizations between what we know about temperate food webs in streams that are forested, in streams that are about the same size, and how this may differ in tropical streams. So this was exciting because we have a completely different set of role players. And when I say exciting, some of the shrimp diversity, I say, is not only interesting taxonomically, but you think about the way that they might interact with resources at multiple trophic level has generated some very cool adaptations and very cool modes of feeding. Like, for example, this shrimp out here, what you're seeing now is they have evolved like just this almost like brush-like features that they could use not just to scrape algae and materials from rocks, but they also, what this one's doing out here is actually filter feeding. Is almost generating like now like this traps or fans to capture materials that are being exported downstream and then just kind of bring them to their mouth. So you can think almost, I mean, if you think about this picture, and if you're kind of like a buggy geek like me, or like an entomology geek, think about almost like a black fly, like having just their feeding mechanisms with their brushes, just capturing stuff as it's flowing downstream, but obviously a lot larger. You're talking about now some macro consumers in this system. Now, what was exciting is that before I got there, one of our colleagues, Dr. Wyatt Cross, who's a professor at Montana State, he has spent some time out there working in quantifying not just the biomass, but also the turnover rate of these varieties of shrimp in these systems, particularly in systems out there that are fishless. And what happens in Puerto Rico is that you have, particularly these sites, sets of waterfalls that limit the movement of fish to very upstream reaches. So you have the systems that now, with the absence of a fish predator, have this very dominant and diverse communities of shrimp. So in fishless streams, Whitecross had measured biomass of shrimp to be about 4.5 grams of estuarial mass per square meter, but also because their turnover rate, their growth and their turnover was so slow, that really resulted in a very small amount of production, so less than 1 gram of estuary dry matter per square meter per year. So that was very interesting because we're really understanding that this organisms not only had an incredible role in the system for turning over resources, breaking down coarse material into small material, but also increasing the retention of these materials in their own feeding, but they're really not turning over very quickly. They will be in the systems for a long time, which was exciting for us because I had a chance to come to this very same systems and now really work on the other component, the other compartment, the macroinvertebrate community, and see how they contributed to now energy flow and exchange of materials. And I want to plug that this was an opportunity that I started working myself out there as a postdoc, but it also was continued by the work of a student that I recruited in Puerto Rico that had a chance to work with me as an undergraduate and as a graduate student, Keisa Rosas, who is now a PhD student at the University of Georgia. So I had a chance to work with Puerto Rican students, get them involved in the work that I was doing as a postdoc, and have that translate into graduate opportunities that fortunately for me, with a student like Keisa, has continued in advanced degrees and now pursuing a PhD. So Keisa did the same type of work that we were doing in Panama, and she estimated the production and biomass of the major macroinvertebrates in the system, aside from shrimp. So now we're talking about now the benthic insect community, and biomass, as expected, was very low relative to the amount of biomass that we saw for shrimp. Shrimp were over 4 grams. Macroinvertebrates, the dominant community, really accounted for a very small portion of that. But however, the production, the turnover rate now, with the smaller organisms that are now growing and developing a lot quicker, is very close to matching the amount that we see for shrimp. So like now they're turning over almost 0.5 gram, whereas you saw for shrimp, they were about like 0.8. So that was exciting for us to see that this smaller portion of the total biomass was really turning over very, very quickly. And even more so for us to think that a lot of these organisms may contribute as they emerge into terrestrial adults to food webs at riparian areas. So Keisa went a layer beyond this. She examined the diets and the contribution of the diets from these invertebrates by different resources, which was one of the very first detailed food web, quantitative food web studies that we've seen, not just in small tropical island streams, but in tropical streams in general. And what Keisa was able to do was take some of those dominant consumers, where we had out here two families, mainly like 2 families and about 3 genera of caddisflies. This is the Calamus rata, Phalloicus, and this is a hydrocycid, and two families of mayflies, and about 3 different subtribes of chironomid midges. And what she was able to do is look at the contribution of different resource types to their diets. And what you're seeing out here, again, not just different resource types, but now the width of those arrows telling you how much material is being transferred across those units. And again, and what was not surprising to us with this very forested systems that are getting major contributions of organic matter from the terrestrial zone, that detritus and organic matter, decomposing detritus and amorphous detritus, kind of like an unknown detritus, was really a large contributor, or plant material was a large contributor to their diets and to the pathways of energy flow or transfer of energy in this benthic systems. So that was really cool because, again, not a lot of studies doing this type of work in the tropics, only a few that do it, but it really gives us the opportunity to think about how now the larger picture of aquatic macro and you know, smaller macroinvertebrates as consumers is really contributing to the transfer of energy in these systems, and how they might also contribute to adjacent ecosystem in terrestrial areas. So there's still questions to be addressed here. Wyatt did a lot of work again with the shrimp as macro consumers. Kesa did a lot of work here with the macroinvertebrate communities. But now we're trying to understand the exchange of those materials and how they might not only like become some sort of like top-down control of resources, but also contribute to resources themselves as they now generate from coarse particulate to smaller particulates that might be transferred downstream, but likewise how this might contribute to larger or higher trophic levels as they leave the stream ecosystems. So this was really cool. We were really starting to provide perhaps like, you know, a driver for more studies of secondary production in the tropics, and particularly in tropical islands, but not just tropical islands, just the tropics in general. And I want to give a plug as I say that even though there are few studies of tropical food webs and quantitative food webs using macroinvertebrates, this has generated now a new community of collaborators and scientists. And in particular, I want to plug the work of José Melo, who's a Brazilian young collaborator, whose paper on the secondary production of tropical streams in the Brazilian savanna just came out, is hot off the press in Freshwater Science. I encourage you to check out that paper. And why it is important is because not just the Brazilian savanna happens to be one of the largest biomes in South America, but it's a global hotspot for biodiversity that is facing critical amount of habitat loss. So the work that Che and collaborators were able to do out here really informs us of how the systems might be functioning as they face threats of not just habitat loss, deforestation, and so on, but also the additional pressures of having now like increased human use in surrounding areas. So Che was able to do the same. I'm trying to plug it out here with the same story for the work in Panama and in Puerto Rico. And what we notice is that the macroinverted biomass in production that Che and colleagues were able to measure out there was much larger than anything we imagined for our tropical streams of about the same size in Puerto Rico and even in Panama. So not only did he find, you know, more than I think, like, well, in a sense, more than 20 times the amount of turnover that we expected to find and that we had seen in some of these other areas. But it was important to note that the amount of material that is being processed by the detritivores in this system, the amount of turnover of coarse particles into fine particles was relatively significant for this system. So we're excited that this might drive new and ongoing projects in the tropics and perhaps new opportunities, as we always look for great students to join our labs, but also to continue a legacy of work in tropical streams, not just with collaborators from Puerto Rico and from Brazil, but from other places that I know we have great freshwater scientists. So it takes me a little bit to my current work. I've been in Southeast Georgia now for about 10 years. My lab here has mainly centered on coastal plain rivers and wetlands, and in particular, rivers that are somewhat in parallel, draining down the coastal plain to the Atlantic, but they have very different conditions and very different set of challenges. For example, the Savannah River, which forms the border between South Carolina and Georgia, is heavily managed. It has 3 very large dams, and some of this for hydroelectric power. Some of them provide some of the great sources of recreation in large reservoirs and state parks in both the state of Georgia and South Carolina. Also, the Altamaha River, which is one of like our treasures, not just for the Southeast, but globally, a system that is largely undammed, and particularly as both its tributaries in the Oconee and the Ocmulgee, which are just above the coastal plain in the Piedmont. Once it goes beyond to the coastal plain, this is a river that has been allowed to experience natural flood poles and natural fluctuation and heavily protected along its riparian edges. Now, of course, there's always the the threat of population growth, particularly as we get towards our coast. But this has been a treasure to work with as a somewhat on-outer system. And in the river in my backyard, the Ogeechee River, where most of the work of my students has occurred, yet another treasure, because a coastal plain stream that is almost entirely in the coastal plain, a very slow moving, a very low retention coastal plain river that has really, because of its low gradient, has been spared in terms of many of the impacts historically that this river's had in removing riparian vegetation and the removing of hardwoods to be transported for construction for many years that remove important habitat in the systems. So I'll plug really quick the work of two of my recent students. One that, if I go back to the things that hooked me to aquatic ecology as an undergraduate in Kansas, one of my students really took a continuum approach in studying macroinvertebrate communities or assemblages from near the headwaters all the way down to the coast to really try to understand not just changes in those assemblages in their diversity or in their richness, but also to try to understand how food webs might change, not just spatially from the up or headwaters tributaries, but as you go all the way down to the coast. And what you see on the left here is this is a connectance food web where all the nodes or the dots from the bottom are resources to organisms or consumers. The one on the left is from a tributary or reach at the upper site or the tributaries, whereas the one on the right is some of our lowermost sites. So you see how the difference in connectance and the difference in the amount of linkages that you see is quite impressive. But he also looked at this more in a temporal standpoint and looking at the same site over time as you move from spring to summer to fall to winter, how much those connections and just the availability of taxa changes as they experience flood poles or as they experience baseline flow. And then another project that I'll plug, this is from another one of my recent students. This is the work of Jose Sanchez, a Puerto Rican student that also worked with us at the LTR in Puerto Rico and moved to Georgia to pursue a master's, currently now pursuing a PhD at Montana State with Wyatt Cross. Again, the importance of collaborators and networks. But Jose, what he did here was he studied one site over a long period of time. We had established long-term data sets, and he was able to examine leaf decomposition over time on a data set of more than 8 years to develop models on what affected or influenced rates of decomposition at a coastal plain river. And his models are suggesting that some of the major impacts that we see would be in terms, obviously, of temperature and how temperature might influence now the breakdown of materials, but also saw impacts on discharge as physical fragmentation of these resources occur, but also on the impact of consumers in our shredders or detritivores or invertebrates that are colonizing these leaf packs and breaking things down. And I'll plug here that on his discharge model, even though we're seeing somewhat a flat line, what we suspect is that this relationship is a little bit more like almost like intermediate, where you have a change, somewhat of a polynomial fit where it is low at low discharge. It really peaks down the middle, but then reduces again as you have very high peak flow. So the cool thing about his work is that it generated collaborations with global networks and allowed it to participate in this global studies of decomposition to try to understand and model these patterns, not just at our sites in coastal plains, but more globally with partners all throughout, not just the US, but the other side of the pond. So that's been exciting as well. Now, we do like to do all kinds of other things. I know that my talk was gonna be geared towards really broadening participation. So I'll talk a little bit about two projects that are really centered on providing opportunities for underrepresented minorities. And I wanna just touch base that this just doesn't happen at the college student stage where we do work, of course, us with undergraduates and graduates, but also with adults and also with children. One of the projects that we're still involved with is a collaboration with middle school and high school teachers. These are all teachers across the state of Georgia, to expose them to freshwater ecology and basic skills in bioassessment that then they could use to develop project-based learning with their classes. So now we have students at high schools and in middle schools generating data sets to be able to now get exposure to freshwater science and the ecosystem functions of their neighboring or their backyard systems at a very early age. I'll also touch back on the impact that the REU program had in me as an undergraduate. So now I've been able to come full circle. I do have, I'm the PI with a collaborator on an REU site on coastal plain science at Georgia Southern University. And I encourage you to of course, tell your undergraduates to come apply and come learn about the ecosystems of at the East Coast, but also for graduate students and postdoc to, if you haven't had a chance to get involved with an REU as a mentor or as a way to really connect in recruitment, I urge you to try to seek opportunities to participate in REUs as a near peer mentor. They're probably more significant than the contributions that we can make as faculty when we interact with the students. And then lastly, as Albert mentioned, for the last 10 years, I've been blessed and fortunate to have collaborations through a partnership with the Society for Freshwater Science and collaborators, not just at Cal Berkeley with Dr. Tina Mendez, who is there, but across different universities across the US, to bring underrepresented minority students to the annual conference of the Society for Freshwater Science and have them participate not just in professional development opportunities and skill training, but also to expand on their network. And the key here is that scientific societies can play an important role in not just exposure to the science, but really providing this network of scientists, potential mentors, potential employers, but in a way that is just not as typical as what we could do in the classroom. So I am very encouraged about the progress that we've made through this program, the InStars program, but even more so that the program, as Albert mentioned, has been now recently funded to expand, not just to provide opportunities for undergraduates, but also for underrepresented minority graduate students and early career professionals interested in freshwater science. And not only at the conference, we used to just bring students to the conference, but now we'll have other components. We'll try to include here a cohort building activity or a river float where we'll get to move from year to year, and hopefully in partnership with another network of scientists that do field river expeditions or field river trips to bring our students together to collaborate, learn about different rivers, but also things that are more even towards the safety measures and not just the science. We of course have our annual meeting opportunities for professional development, but we've also partnered with the National Ecological Observatory Network to provide training workshops on statistics, things like R. We've partnered with folks at Virginia Commonwealth with one of our collaborators to provide workshops on visual communication, dissemination of data, presentation skills, and also building this online community. And if we know now that the ability to do this virtually is critically important, obviously we try to generate these opportunities and these networks to expand our reach on perhaps a virtual space for the scientists. And again, I'll plug my collaborators, Dr. Amy Roseman and Amanda Rugensky at University of Georgia, my good friend and colleague, Tina Mendez, who's in the audience. Tina and I have been working on this since day one for over 10 years. And even before we were academics, maybe as we were just getting started and spreading our wings as InStars, Dr. Dan McGarvey at Virginia Commonwealth, and of course myself. So I do encourage you to look into these groups. And of course, things are different now. We used to do this at conferences. We've partnered with the Consortium of Aquatic Scientific Societies to provide this programming and exposure to this at conferences like the SACNAS conference, the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science. This is what it looked like maybe 5 years ago. This is what it looked like this year. And of course, with other scientific societies like the ESA and the Society for Wetland Scientists, we've really expanded on this outreach to really get folks to understand the value of not just scientific societies, but also broadening participation in STEM. And with that, I really appreciate the opportunity. Like I said, I'm long-winded. I think it's, it's 7:30 here, 7:23 has got to be, what, 4:23 out there. So thank you so much. And I'll be happy to address any questions or any concerns that you may have. But thank you for having me. Yay, virtual claps. Very good. Thank you.
Thank you, Checo. This was really great. We have time for maybe 2, 3, or 4 quick questions. You can ask them or chat, and then we'll give you.
Yes, I see. And thank you to Charlotte. Yes, you're very, very fortunate to have Tina. I am very fortunate to have met Tina. And again, and through the Society of Freshwater Science, we've not only built a great friendship, but this network where we've had the opportunity for us. I mean, I think Tina and I will both tell you this was us. Well, I won't say decades ago, but the opportunity to see students grow through our InStars program, and now know that they are in graduate programs, postdoc, and some of them already at the academic level or in professional positions as scientists is incredible. Thank you, Denise. Thank you.
I have one question.
Go ahead, Kyle. Yeah.
That's okay. You've been to so many different systems, and they all seem like you could come up with an exception to the river continuum concept in each one, most likely. And I'm wondering if we should be teaching and thinking about larger theories and frameworks as a jumping off point rather than like a truth to be tested against.
No, I mean, I absolutely. And I think that should be our goal. I think a lot of times, and I think from a teaching standpoint, when I teach a river ecology or a stream ecology class, and we introduce this well-known frameworks like the river continuum concept and the flood post concept, I mean, I always feel that it's critical that we challenge our students to understand, right, that the framework is the framework. I think about it as like, okay, not all river systems are gonna exactly fit the picture that you see in the paper, but the concept or the premise does fit, right? For me, I tell my students the physical is driving the biological. When I look at a prairie stream, and perhaps their tributaries are not heavily forested as we expect, and perhaps the rivers I see now going from like the Piedmont and the Appalachian all the way down to the coast. But the context of like less riparian buffer driving higher primary production will possibly drive more resources for shredders and for primary consumers. So that still fits. It's just it's fitting perhaps at a different place. But the physical conditions are really what bringing that, as Vinod would say, harmony across what we see from the resources to the consumers. Flood pulse concept, same thing. I got to think about it now that the flood pulse concept, I have to add the potential impact of hydroelectric dams, changes in the predictability of the flood pulse, but think about how that driving or that connecting of resources in the main channel into the floodplain and how that may come back is really a driver, not just for production, but also for the way that organisms might even adjust or adapt to their life history over many, many generations. I think about systems now that are not flooding, right? And how that might impact the turnover of taxa that have predictably used that system for years or so to develop to a particular stage. Now, if those systems are not being flooded for some time, that's going to be an issue in having those organisms come back. So I think, we always got to challenge the way that we see the framework into how the conditions might apply to the systems that we're seeing now. And of course, add to that, if you have an interest in restoration or an interest in disturbance, think about how the framework might allow you to make decisions on the application of restoration to really be able to provide the resources or the functions that were there before. Does that answer your question? I'm long-winded, not just in the presentation, but also in the answers.
I have a question meanwhile, if you can think about any other question and put it in the chat. I really like that food web topology that you showed, that how it would change over time across seasons in the Georgia rivers. And I was wondering if you asked any similar question in Puerto Rico, given how much knowledge you have about the functioning, like secondary production and trophic transfer efficiency. Like, are these stable over time? Because in California, we always think that every year there's a reset with the rains in the winter, and then who knows what happens. But I can imagine that maybe Puerto Rico is more stable.
Well, I think that's a good question to ask, particularly in terms of what are the drivers, right? So obviously, we had when these studies were happening, we had a period of time in the island where we were really worried about drought in these systems. And we were really targeting this baseline data sets to be able to ask more questions about the effects of potential prolonged drought. And this was, you know, everybody, this is an LTR, right? So everybody was coming together thinking like, hey, we need to get ready for this renewal, and drought that's gonna be, everybody get on the same page, it's gonna be drought. Then a little thing called Hurricane Maria happened a couple of years ago, and it really, you know, impacted our thinking, particularly, I mean, as somebody, I grew up, spent my first 18, 19 years in Puerto Rico, and I probably saw, you know, to memory, maybe one major hurricane in the last couple of years. That has changed quite a bit. And when these hurricanes happen, and we're lucky enough that a site like the LTR had great people generating data back in the late 80s, early 90s when Hurricane Hugo hit that site, and to understand how that influx of the amount of material that gets input into the stream, how it might overwhelm the system in terms of being able to turn it over. So there's great people out there, including Alonso Ramirez, who was my postdoc advisor and still collaborator, and plug. He's also very involved. He's the president of the Society of Freshwater Science now. We still have a network of collaborators that continue to monitor the systems. The work on the shrimps in that area has you know, expanded more than 20 years of continuous monitoring. It's resulted in great education and outreach films, a film by Freshwaters Illustrated. That's the picture that I had in there. So our goal is, like every LTR, to try to understand and use these data sets to drive more studies. I happen to be interested in disturbance, so I think, as going towards your question, Albert, I would like to know how variable secondary production is when you have like this variability in you know, disturbance, this variability from very dry years to very let's call it strong or active hurricane years. The challenge with that is that secondary production, as you know, and Kyle, as you're learning now, this is a lot of work, and to be able to collect data to generate these estimates of production, you really have to embrace the challenge of continuous studies and long-term studies. And there's definitely an interest in doing it. It's usually our funding cycles, as many of us know, tend to be challenging to have studies like that. So an LTR site provides that opportunity. I remember as a graduate student, hearing from actually one of your colleagues from Mary Power, telling me as a young graduate student, hey, sometimes you just gotta continue your projects, even if you're just kind of scratching nickels together, just to make sure that you have the opportunity to answer these questions at the long term. And I'll never forget that, right? Like, just the opportunity to think like, okay, we could keep collecting data. Perhaps this will allow us to answer questions that are bigger than what we could do in a single project.
Yeah, thank you. This is really interesting. I think we'll wrap it up, but just for those of you who would like to stick around, we'll have some informal chat with Checo now for the next 15 to 30 minutes. So if you just want to ask questions or chat, no recording, more informal atmosphere, just stick around. Thank you again.
Thank you all. Thank you for having me. Love to chat some more.